Hello, and welcome to the latest episode of Our Future in Space. My name is Dr. Jeff Greenblatt. I am your host and the Vice President of Science and Research at Orbital Assembly Corporation. In this podcast, we explore the technical, economic, social, political, and ethical considerations of sending humans into space, not just for short periods, but to thrive long term throughout the solar system. We now have the ability to sustain humans off Earth in low Earth orbit, and very soon, anywhere we please. So I hope you enjoyed this latest episode, and I am joined today by my co-host, Eric Ward, who's the Vice President of Engineering Design at Orbital Assembly Corporation. How's it going today, Eric?
Oh, it's going pretty well. I'm excited for the podcast today. We've got a guest today, Dr. Kate Krueger from Helikon Consulting, where she's the founder and CEO. She's passionate about using protein science for food innovation and has extensive technical and field-specific strategy, including 3 years as research director at New Harvest, patent contributions to Perfect Day Foods, and a PhD in cell biology from Yale University. She also worked as a technical expert at XPRIZE Foundation. Kate, welcome. Thank you for coming. So, can you tell us a little bit about the world of synthetic biology and why it should matter for space?
Yeah, definitely. Thanks so much for having me, Jeff and Eric. A little bit of background on high-tech foods. As many people know, if you head into space, food suddenly becomes a consideration. Basically today, what I'm interested in talking with co-hosts about is not only using food in space, but also using different types of synthetic biology for innovation as people think about what it might look like to live in space long term. So when we talk about that type of innovation, what we're talking about is a variety of disciplines. We're talking about synthetic biology, the ability to make biomolecules, biopharma, biomedicines in space. We're talking about the ability to make proteins. Molecular catalysts for various reactions in different industrial applications. We're talking about food, we're talking about food proteins. So how do we make things like milk and eggs? And one of the most complex things, how do we make meat? How do we think about making a tissue? How do we think about any kind of mammalian cell growth that we could use to sustain ourselves, similar to eating a steak or a chicken? So that's a whole lot. It's really complicated. We start heading from tech into deep tech, and I'm really excited to share this all with you today.
Oh, we are too. No, we're so excited to hear. And I have to say, Kate, I first reached out to you a few months back because I had really just discovered this world of cellular meat production and synthetic biology more generally. And for me, even somebody who follows high tech and the space industry for a long time, this was kind of a revelation to me. So I wonder if you could just start our listeners off with a real basic idea about what even is this and what's possible today, and where are we headed?
Yeah, definitely. So this is a really exciting field because we are way farther than we were 10 or 15 years ago. So we are seeing developments that have come very quickly. So when you're from a background like myself that's a little bit more biomedicine or traditional science, what you tend to think of as fast innovation is within the past 20 years or 50 years. And that's true. That's very quick, I think, in many senses. That's a lot of innovation in what's less than a human lifespan, but it's absolutely nothing compared to the speed of food innovation. There are a lot of drivers for that. One is the regulatory process for food approval is very, very different from biomedicine. The other is many of these new technologies from the biomedical and bioengineering world have simply not been applied to other disciplines such as food. And so because of that, within the past, even just 10 years, we've seen the adoption of things like the Impossible Burger. The Impossible Burger is not cell-based meat, so you're not growing whole mammalian cells in a culture. But that said, you're doing tons of science to make the Impossible Burger work. So what may not be obvious from eating a burger at, for instance, Burger King is just how much science goes into that burger. So when we're looking at the Impossible, what we're looking at is what's called recombinant protein production to make a protein called leg hemoglobin that is colored red for your burger. So to do that, you're making tons and tons and tons of protein where you've taken the DNA from one organism and put it into another organism to produce a protein.
Gotcha. All right. So this is what some people have sort of labeled. I hate to use the word, but it's out there. Frankenfood, right? GMO, sort of this notion that you can take the genetic information of one organism or just a snippet of it and put it in another for the purpose of expression, right? Which is a very useful thing for us to do. But some people are kind of afraid of this, whereas I think others really understand its benefits and its safety. But sorry, but go on. Yeah, I think
exactly. And yeah, I think that technology is really where a lot of this promise lies. There are words we don't use, so we try not to use words like schmeat, for instance, or other derogatory terms that don't need to be derogatory. I think they're kind of funny, actually. But yeah, I mean, that process you're describing is genetic engineering. It makes what we refer to as potentially GMOs. Not all these products are necessarily GMOs. Sometimes you can just engineer an organism to make a protein, which is in and of itself not genetically modified. So it is important to make that distinction there. Also with recent genetic technologies that are very, very delicate. In some cases, you're not ticking the GMO box there either. It's more like lightly massaging biology to get what you're looking for. So yeah, even that, I think what's cool is we're pushing the innovation boundary in terms of what we can do with high-tech science without triggering GMO concerns. That's just a little context there. I don't know if that's useful.
Yeah, it definitely is. I think some people are sort of already wondering, well, what really is this? And also it's changed so much from what was first, introduced into the market, 20 years ago or more.
Exactly. Yeah. And, some of it's GMO proper, some of it's not. A lot of it's not. And that's really, really exciting. But that said, the very fact that we can think about doing genetic modification or not doing genetic modification at a big level is very, very exciting. And, if you start thinking about what we've been able to do with, even more broadly synthetic biology, what kind of high value molecules we can make that are really, really expensive. So things like cancer therapeutics, for instance, or even vanilla. People don't realize vanilla is really complicated. So vanilla is made by a very specialized plant that only grows in certain parts of the world. It's very expensive. But we can use biology to make it much more inexpensively in the lab through the method you described where you put DNA from one organism into another. So it's very exciting.
Wow, there's a lot to unpack here, and I don't want to run too fast. I mean, I know that we're all us on the call are fairly familiar with this, but let's just slow down a little bit and talk about what. Let's take the example of the high value cancer drugs. So what we're talking about is something which is difficult, if not impossible, to synthesize in a chem lab, but we can use biology that already knows how to make this naturally if it's given the genetic instructions. And what are we doing in that case that makes it exciting?
So for things like that, there are kind of two different classes of molecules that we're able to make that can be used for instance, things like cancer therapeutics. So one is what's called small molecule production. Another is what's called protein production. So small molecules are things you could take in pill form, at least in many cases. And what it takes to make those is oftentimes, if you're making them biologically, what people are doing is they're engineering entire genetic pathways to produce novel molecules, or they're tweaking existing pathways to make more of a certain byproduct that's really useful. So that looks like a whole lot of essentially engineering of very complex systems, one molecule that makes another that makes another. So imagine the most complicated DNA or type of chemical synthesis you could do, for instance, in organic chemistry labs. So think about doing that over and over and over again. So maybe 12 or 15 steps, each of which need to have very specific steps and very specific molecular tuning to make things happen. So that's one type. And then another is how you tweak cells to make very complicated, what are called large molecules or proteins. So that's injectables, other types of therapeutics. So, and that's a whole nother type of kind of genetic massage for a cell type to really get what you want.
But it's important because of the obviously the human benefits, right? I mean, that you're talking about saving lives in this case, and there is money available for doing this. So even if it costs, I don't know, what are we talking, like $1 million a kilogram for some of these materials, maybe more, but that kilogram goes a long way because you're really only talking a few micrograms of this material to be active. I'm guessing here, so please correct me.
Exactly. And so that's the thing that's so exciting about this tech, right? Is we've gotten to a point where we can do it, and we can do it for a very high price point. But we've gotten so much better at it that now we can start thinking about what it would look like to do with larger quantities at a lower price point. And that's what it looks like when we hop into the food space. So all of a sudden, the technology is not necessarily harder. In some cases, it might be simpler, but you've got to hit, somewhere approaching price parity with milk or eggs or beef, which they're already very optimized systems made by complicated organisms. And on top of that, they're government subsidized in many cases. So everything about that process is very hard from a volume and cost perspective. Space is a really cool example because it runs counter again to some of those variables, right? All of a sudden, you don't need quite as extremely low price point if you have a better production system that works better with where you're operating, right?
Yeah, not to mention not having to bring cows and chickens with you to space, right?
Right. So yeah, I mean, I think that was part of our earlier conversation, Jeff and Eric, that was so exciting, was talking a little bit about like, okay, which assumptions about food production are actually turned on their head in space? And one of those is cost. And the other, of course, is exactly what you're talking about. It's the various complexities of having different ways of making food in space. Are you going to levitate a cow, and stick that up on a spaceship? Or are you going to think of instead about, okay, what's a bioreactor going to look like in a space context?
Right. And we're really talking about is we need things that can work in very small spaces right now. I mean, eventually we hope to have plenty of room for people to live, and it will basically be like floating cities, and you could imagine pasture land, but still that land is at an extreme premium, and we'd rather use it to build apartments and maybe a nature park than to have cattle grazing. Right.
Yeah, there's nothing more unattractive than a massive factory farm, especially if you have low square footage.
Yeah, for sure. So, okay, so I think I understand the case for space, although we'll dig more into that. But just take a step back for half a minute. So, why are people excited about making these synthetic foods, for Earth consumption? I mean, I have to ask the obvious. Why would people want to do that instead of just continuing with the systems that we have?
Yeah, I mean, I think that's a great question. So there are a whole lot of reasons people are excited about it. I think some of us are from a technical background, scientists by training, who just think this is really exciting. We can even think about doing this. So the ability to tweak these molecular levers is around now. The technology is established and we can put it in new places. So some of us are just really, really excited about the prospect of doing this at all. I think there's a lot of drivers as well that make it really worthwhile to consider pushing this tech quite hard. So climate change is a huge one. As we start looking at, rising temperatures, thinking about what we can do to pull down those costs is really, really important. And the interesting thing is it becomes really clear that reduced water and reduced land footprint seem to be, in many cases, pretty clear results of a reduction in animal agriculture and an increase in things like cellular agriculture based on preliminary modeling. However, energy consumption is a little bit more of an open question, but we have so many other technologies to mitigate that. That's kind of something I think we can table for much of the discussion.
Yeah, you mean from a climate change point of view, right? It might require more electricity or heat, but we have wind and solar and geothermal and
Exactly. You're
carbon.
going to need to link some technologies together to be 100% sure that you really are ticking the environment boxes that you want to tick. But rather than having whole organisms, it's possible to think about what ticking those boxes looks like in a really quantified way. So
And I suppose too, yeah, you can engineer around those boxes where if you're using cow meat, you can't very easily make a more energy efficient cow, where you might
Right.
be able to spend development time to make a more energy efficient bioreactor.
Right. You can't use nuclear power to power a cow. It just doesn't work quite that well.
It doesn't work.
Yeah. Although people have definitely, I mean, we kind of already did that, right? We made our livestock, species more efficient, right? I think that
Exactly.
For
We've
breeding, yeah.
optimized them
chickens grow
to
a
an
lot
extent.
faster than they
Yeah.
used to, and they probably lay more eggs, and the cows produce more milk and grow faster for meat production. But we're kind of at the limit there, and we're seeing the
Exactly.
environmental downsides.
Yeah,
Yeah,
we've
not to
really hit
mention
these
land use, which
biological
is
limits. Huh? Sorry.
sorry, say that again.
Oh, we've really hit biological limits, just to reinforce what you were
Mhm.
saying. It's very, very
Right,
true.
right. Yeah, so maybe
Well,
just walk us through. Oh, sorry, Eric, were you gonna
oh,
go ahead?
I was gonna say, we've talked about a lot of the more kind of, arcane aspects of this, but I'm curious, and I'm sure some of our listeners are curious about cellular meat. Is it healthier for you? How does it taste?
Mm,
Like,
yeah,
tell us
good
a little
point.
bit about that, and maybe how it's like, what that process is, how it's grown. We all kind of understand how a cow is raised, but what would we be eating? How healthy is it? And how would we use it?
Yeah, I mean, so there's a lot of questions there to unpack, and I'll start with some of the first, which unfortunately are the least answerable, but it's pretty quick to do that. So the first is taste. Very hard to say. No products on the market yet. So
Mhm.
taste is an open question, I'd say in general. Of course, very, very important to companies. Still, until we have a product on the market, it's really not possible. You can't, for instance, go to Burger King and get an Impossible Nugget and say, okay, this is what it tastes like. So very much definitely a TBD. But that said, segueing into your next question about process, the options are much broader when you're talking about what is possible from cellular agriculture than is what is possible from an animal. As Jeff mentioned, you really do hit these biological limits with what you can do with a cow or a pig or a sheep. And we've gotten there, right? We've optimized the price down as far as we can go. We've created things like Wagyu, where we got very delicious types of meat. But there's not a whole lot more we can do. But that said, when you go to the extreme of creating meat in a bioreactor and you've done all the bioengineering necessary to make that possible, there's no reason why you can't tune flavor, taste, parameters pretty endlessly beyond standard imagination. So one example of that is some of the researchers at my previous organization, New Harvest, were working on growing meat cells, for instance, on various types of what are called scaffold materials. So things like where you'd be growing animal cells on a piece of apple, for instance. So you could create an apple meat hybrid in a unique way. That's just one example.
Whoa.
So you can get like high fiber meat, essentially.
Okay. With extra nutrients, I guess it's sort of like a
Yeah.
complete meal in one bite, sort of.
Oh yeah. I mean, like
Huh.
that's the thing is right to the process, it's very complicated, right? So you have to take a tissue biopsy from an animal, grow those cells up into a standard culture, make sure they're optimized for growth. So that could involve things like genetic modification, for instance, very highly tuned ingredients to feed the cells, etc. And then those cells are grown in a bioreactor either with what's called a scaffold material or without what's called a scaffold material. And then a scaffold material is essentially something that cells can bind to that allows them to go past standard limits of cell growth. So cells can only stack so far on top of each other before they hit the oxygen diffusion limit and some of these other really key growth parameters that are limiting if you're thinking about making a thick steak. Those are not such a problem if you're thinking about growing very thin sheets of cells or single suspension cells, for for instance, in a growth bioreactor, and then creating something like, for instance, ground beef or something.
Okay, right.
So
Easier to make.
that's a
Yeah.
whole different level of complexity, whether you're going to make a ground beef product, or you're going to make something that's called structured, or a little bit more like a cut of chicken or a fillet of fish. But yeah, if you're going to do that, you can play with the genetics if you're already genetically engineering things. So you could increase the health parameters by engineering in a genetic pathway that makes antioxidants, for instance, or makes all sorts of other compounds, flavor compounds, for instance. You can tune what you feed the cells to an extreme molecular level of control. So what kind of fats you put in? Do you want to use omega-3 fatty acids with beef, for instance, and get the health benefits of salmon potentially while you're eating beef? No reason why you couldn't do that, as long as the cells can tolerate the difference in feeds. But yeah, the options just become very, very wide. So it's really exciting what you can think about doing.
That's
Yeah,
really
there's also a uniformity, I'm guessing, too, that you can produce something which is exactly the same shape, color, flavor.
Yeah.
Right.
Exactly. And you don't have a lot of the standard concerns, right? So cell culture by necessity needs to be very sterile, so no contaminants at all. And because of that, whatever product you'd harvest downstream has to be essentially germ-free. So you don't have any concerns about, salmonella or, a lot of these other pathogens, as long as packaging is done appropriately. So that's an entirely different set of considerations.
But another plus, yeah, for sure.
Yes, you mentioned a second ago feeding it with omega-3s. What in general do you feed a bioreactor with, right? I'm thinking we're making these in space, right? And maybe we're talking about an O'Neill cylinder of some thousands of people that this synthetic biofactory is feeding these people. What kind of inputs does that factory need in order to grow this meat?
Yeah. So that's a really good question. So the great thing about it is so I'm a Soylent fan, I will admit. And if anyone here on the podcast has tried Soylent or is familiar with it, I'll give a brief recap. But essentially, it's a powder that you can reconstitute with water to create a meal substitute. And Soylent is great because it can be fit into a very small volume due to the powder form. Cell growth medium is largely like that. So it's a lot of powdered ingredients that you could reconstitute in water. But if anyone's tried powdered Soylent, they'll know it's not quite the same, for instance, as maybe even the liquid Soylent, which I find vastly better. I'm not exactly sure what technology goes into making that different, but my guess is in part it has to do with lipid emulsions. And that, at least for many cell types, is the case. So, for instance, if you're making up a batch of insect cells, a lot of the ingredients you put in there for the cell growth media, like Soylent, is a powder. So it's things like amino acids, it's sugar, it's various what are called hydrolysates from yeast, for instance. Those are super standard ingredients. But you also will need something like a lipid emulsion. So you will likely need some kind of a situation where you do need to bring some liquid up and possibly have some processing step. At least that's standard for many cell growth medium manufacturing techniques. So it's not completely powder plus water, but it's pretty close.
Well, that's okay. I mean, in any place that we're going to have humans living for long periods, there's going to be plenty of water, that will be recycled most likely, and small amounts, supplemented, what gets lost. But yeah, having access to water, especially if just part of a process loop, should be not a huge issue. But yeah, but these other ingredients that I take it that these are not that hard to either make or keep keep nice and fresh while you're waiting to grow your tissues.
Those are pretty straightforward. So lipids do have a certain type of shelf life. So that's a consideration. Price point is, of course, also a consideration because, again, getting any of these technologies remotely near food pricing, even on a space level, is a bit of a consideration. So that will be a challenge. Is okay, how do we get the amino acid pricing low enough? How do we get the hydrolysate pricing low enough? Those things are going to be questions for a bit.
So Tom Klein was wondering that he had a two-part question: what do we feed the bioreactor, but also where do we get it? Like, where do these lipids come from? Where do these amino acids come from? And in an Earth-based process, right? Maybe we've got some industrial supply chain in the world that's like, this is a byproduct, but where do they come from here, and where might they come from on a more kind of contained environment?
Yeah, I mean, I'll answer part of that question. I think part of that question I'm
Yeah.
going to have to put back at you two, which is what you think we're going to have access to in space or in the types of space environments you're talking about. So you'll definitely need some things that have to do with Earth byproducts at this current stage in time. And that's part of what we're looking to get rid of. So there are two major components that we need to work on. The one that everyone talks about, which is cell growth medium ingredient called fetal bovine serum. So that's a very expensive media component that contains a whole lot of protein and fat components that is often added to cell growth media for mammalian cell culture. The good thing about getting rid of what's called FBS, fetal bovine serum, is the cell therapy industry is also actively working on considerations around this. So that's a separate biopharmaceutical discipline that, much like cellular agriculture, they're also looking to produce mammalian cells in vats, just human mammalian cells at a much higher price point to make human therapeutics to essentially cure cancer. Again, so they're working on this problem.
A lot of fascinating crossovers here for sure.
Yeah. But I guess the question I'd throw back at you all is, what do you think we'll have access to in space in terms of infrastructure?
Yeah, that's a great question, and something, of course, Jeff and I have been talking a lot about in this context, but also working on in the more short-term context at Orbital Assembly. But in large part, the way I think of it is that when something like an O'Neill cylinder gets set up, and you have this space-based colony, I would expect trade between different space-based colonies and planetary colonies like Earth or Mars or the Moon. But in general, you kind of want to think of it as a closed system. I mean, in a way, and people have been talking about this, I think, since Carl Sagan in the 70s, that Earth is a spaceship, or we're living on spaceship Earth, and in a large way, we get sunlight in, and we radiate television emissions, right? But it's a closed system. It's a very large one. It has a lot of capacity to buffer changes, but some of the climate change that we're looking at are humans kind of reaching into that buffer. In a something like a O'Neill cylinder, you're also this contained system. You've also got sunlight coming in. Maybe there's some trade back and forth, but as you get more and more O'Neill cylinders and a higher percentage of people living in space, you have less of a capacity to just use up Earth's buffer in order to generate or consume resources, and so you really need to have a closed system. And so that's kind of one aspect of it, is this thought of recycling not as something we do to like check that green box, but as a necessity. And so in a way, we can kind of imagine, well, what infrastructure might you have, but that question really becomes, what infrastructure do you need to have, right? If we can't get fetal bovine serum from cows because we don't have access to a bunch of cows anywhere, what kind of growth medium can we have? What are the outputs of this system? How do we recycle human byproducts and clean that and turn that into input processes? And so I think like Jeff made a really good point. We're not going to be having a lot of pasture land, right? We will have a nature preserve land that is good for human mentality, but we can't afford to spend a lot of extra resources just getting one step in the food process, right? So our resource at its core is sunlight and the work you can do with energy. And then, how do we close that loop?
Yeah, you want to keep all the atoms in the system and reuse them infinitely is kind of the ultimate goal. I mean, they're, given enough energy and space and time, you can do that. We already do that on Earth in terms of, going after very small amounts of rare elements in the Earth's crust and, geologic processes then turn around and reconcentrate it or have over hundreds of millions of years. We won't have that kind of time in space, but we'll have more energy. So hopefully
Yeah.
we can accelerate these processes. But
That reminds me of anecdotally the aluminum mining in, I think it was the mid-century 40s or 50s, where it became profitable that garbage landfills were ore bearing in a mining sense for aluminum, where it became profitable to scavenge aluminum from a landfill and reuse it rather than mining new aluminum. And so I guess that does a terrible job of answering your question, but because it occurs to me as I kind of ramble a little bit, and you asked that question of, well, what is available, as a synbio person? I think maybe the answer is too complex to just noodle out, because what you need is an entire closed system, but it's a very complicated system of clothing and food and water and like all these different activities that humans do. And how can we create systems that use each other's byproducts such that it is a closed system without necessitating a huge buffer to put our waste into or something?
Yeah, this question makes me really happy, because I think there's a lot of places we can think about if we're going to go all the way back, right, to thinking about how we solve the food problem. And one thing I'm now curious about, which I'm going to ask you both a little bit about, is kind of what capacities, what kind of resources would be easy to get in space. So one potential input for some of these systems to make high cost reagents for things like cell growth medium is something like an algae production system, for instance, where you could use light to make either fats or proteins, usually fats, but some algae can also be used to make proteins. What do you all think that the capacity will be on a system like yours for capturing light?
Oh, yeah. Well,
yeah.
Eric, you want to start with
I
that?
would say very, very, very large. Like capturing light is in general one of the big, the big things we spend a lot of time developing technology for and thinking on when you design any habitat. Like Jeff is showing up here the Voyager station, which is one thing that Orbital Assembly is working on, and we're looking at it from the anti-sunward side. And so we can see these modules around this big ring where people would be living or equipment would exist. On the opposite side of that, with the sun in the in our background, but the face of that's looking at the sun, 100% covered with solar panels, capturing the most amount of sunlight possible for a given surface area, right? And when we talk about some of these deeper concepts, like we had a conversation with Pekka Janhusen about a colony of colonies, in a way, a big settlement of multiple O'Neill cylinders with a specifically designed mirror to capture sunlight to feed all of those communities, right? And so
enormous
yeah,
mirror.
exactly, because like we said, that's one of the primary inputs. We don't have extra atoms banging around anywhere. We've got sunlight, and we need to power that. We there's even a limit to nuclear power, right? Like that can solve a lot of problems globally here on Earth, but with a colony in space, you don't have a place to go mine and refine plutonium or uranium for nuclear reactors. So the ability to capture light is important, something we give a lot of thought to. You mentioned, for example, algae farms, and like that's also something that that there's been a lot of research around, of using an algae farm to either directly capture sunlight or to use redirected or regenerated power, essentially, to feed that, or to, I guess, power that molecular conversion engine, in a way, right? Because those fats that the algae produce, those atoms don't come from the sunlight, right? And so I think what you're looking at is like not a physiochemical, but a bio. I'm spacing on the other word, but like recycling and regeneration system, where people propose using human waste to feed algae, which produces oxygen to
Bioregenerative system,
breath.
maybe.
But there could be also rather than using the waste of CO2, using other human byproducts to with a synthetic biology, I guess, designed algae farm to convert that back into the input of the process.
Yeah, definitely. And I think, yeah, what you're hitting on there is if light is really such a resource, I mean, you can think about how we make these high value ingredients through other means. So if you are thinking about using light to make something like algae, and you've got urine for your nitrogen source in part, you've got solid waste, right, that can be eaten by things like fungi potentially to produce kind of a rough protein, or something like that. Yeast can eat a variety of things too. Plants can eat solid waste, of course, too. Between all those components, you've got a system where you could very well use a lot of different components that we already have. I mean, the question I think really then is, if you're working with this closed loop system and light, how much CapEx are you willing to put in? Because it does get rather expensive.
Yeah, and here I just want to say, we have to get there. What we're talking about initially is something that would probably be hosted on one of these early space stations, such as Pioneer Station, and it would be growing a little bit of food and would probably have to have some of these growth factors supplied from Earth. But once we have more space, energy, and frankly, capital to be able to invest in these systems, you could then imagine all of the components being grown on the station using recycled waste, just as you're describing. But there's still how much of the station's volume would be taken up by that? I don't think we know yet. I don't know that anybody's sat down and done that calculation for a completely closed loop system. I mean, I know that in prior decades, I mean, O'Neill came up with the cylinder concept in the 1970s, and people quickly took that concept and ran with it and looked at what a completely closed loop civilization would look like, but using the existing kind of traditional agricultural technology at the time. So since then, we've gotten a lot better at understanding how you can grow things in small spaces. And I'm going to bring up an image here of vertical farms, which is another kind of related technology that is less dependent on synthetic biology, but or maybe I'm wrong, and actually there is a lot of synbio in this as well, Kate, if you know anything about it. But it's another ability to grow large amounts of food in a small volume on Earth. Again, to address
definitely. And there's so many interesting overlaps, I think, between vertical farming and between synthetic biology, right? Because again, it's a system where you've really changed the game on yourself, right? So you took something that had a lot of parameters that traditionally we're not that interested in with food, right? Which is square footage. You think about the US, we're not really that interested in really using every square foot the same way maybe someplace like Amsterdam would even, because it's just our parameters are very, very different.
Right, land is cheap basically in the US, most places.
Right. It's just such a different game. But all of a sudden, right, if you tweak that parameter and make that the most expensive one, things like customized nutrients are not a problem. And so, and that's a little bit what we're dealing with here, right? If, all of a sudden you're thinking about customized nutrients, or you're thinking about pulling genetic levers to get you what you need. And all of a sudden those genetic levers, like in vertical farming, could make a lot more sense. It's just all about what you're working with. So all of a sudden you have to be creative in new ways. Robotics makes sense, right? In unique ways it didn't before.
If a human costs a huge amount of money to keep on station, we only want them there to do the jobs that absolutely humans have to do, and probably picking strawberries is not one of them. So we'll have a robot.
And you've changed the game, right? So suddenly robots are better than people for exactly the reasons you're stating, and even more so, robots are sterile. People forget that humans are biological organisms covered in organisms. So we're not that clean. It's very hard. You can put us in bunny suits, but we're still people. But a robot can be, swabbed down with alcohol and whatever other good stuff. And it actually is sterile. It's never going to pick its nose.
Yeah, doesn't have a gut biome.
Get a cold. Yeah.
It's
Yeah.
great. Yeah, exactly. We're just living vectors, whether we want to be or not. I think we all know that post-COVID.
Yeah.
Spoken like a true biologist. So.
Yeah.
Yeah, I mean, and I think that raises so many other questions for a space station, right? How do you change
Yeah.
that game and make it a situation where humans are more safe than we are on Earth, and we're breathing on
Yeah.
each other is less of a problem than it is down here.
So speaking of that, Aiden had a question in chat here. We're just talking about tweaking some of the parameters, right? Humans become expensive. What about square footage? Maybe energy is one of the cheapest aspects rather than other things. He was wondering about space in particular, and if you can speak much to the actual act of genetically modifying biology. Would that be easier in space, in a zero gravity environment, or other factors in space, or is that a really something that's important to have done on Earth and then ship the knowledge up to space? Is gravity important to this? What about that kind of aspect of those sides of the coin we're talking about now?
Yeah, so that's a really, I think, cool question, because it does turn out that gravity does affect a lot of biological systems in ways we don't expect. NASA's done a lot of research around this, especially with things like, for instance, developmental biology, where we get rather complicated processes going on, some of which do turn out to be determined by gravity, which is really interesting. Definitely worth a read if you want to go down that path. That said, there hasn't been a whole lot of research done about growing meat in space. There was a landmark paper back some years ago on growing goldfish cells in space, which seemed to be doable. So that is one of the few cases out there where people have tried this in space. But yeah, in general, the incentives are not necessarily aligned for heavy-duty space research around cellular agriculture just yet. But with recent funding in this space from the USDA and other groups, I think that could be something that might change really soon.
Interesting.
Yeah. Well, plus,
go ahead.
Yeah, I was just going to say, orbital assembly is hoping to change that equation as well by introducing gravity as a normal component of any space-based existence. And so, you'll have as much gravity as you need depending on the application, rather than trying to conform everything to fit in a microgravity environment.
Yeah.
And that could be so helpful. I mean, that could be a real game changer right there. I mean, I think humans ourselves were so heavily reliant on gravity, it turns out, for so many things. A lot of, I think, complex biology likely will be as well. And it's just so nice to have some control over parameters we understand on the ground. That likely any kind of gravity system you can give us would be better than nothing. So that's, I think, important to keep in mind.
So I'm interested. Maybe you could dive in just a little bit more for our listeners. You mentioned that there are some differences in biological growth in zero gravity. We don't know a lot about it, but there's a quote fascinating read in there. We'd love to, if we can get a hands on a paper or a link to put that in the description for the video once it gets posted. But could you talk just a little bit about what those are, to give our listeners an idea of what sort of things could change in
Yeah, so I'm going to give, I think, an example of something that's really cool that you can definitely, I'll send a couple links over, that I think is a really cool case study about growing biological organisms in space. And that has to do with the tomato. So the tomato is one of the most well-researched organisms grown in space, thanks to Project Veggie at NASA, which is currently headed up by Dr. Joya Massa. And one of the interesting things there is the number of challenges that researchers have run into growing tomatoes in space, all the way down to the chemistry of space stations. So it turns out that space stations are full of a molecule that turns out to be a very important plant hormone that controls what's called plant abscission, or how plants die. And that is something called, oh my goodness, my brain, polyethylene.
Ethylene.
Ethylene. My goodness, it is a little early out here. It is ethylene.
Polyethylene, the
Thank
plastic,
you.
but you mean
Yes,
just
that is a
ethylene.
plastic.
Yeah.
Thank you so much. Oh my goodness.
Ethylene is a
got there.
ripening agent,
Morning.
right?
So ethylene is a gas that turns out
Yeah.
to kill plants in excess.
Mm.
And so unless you scrub a space station from ethylene, you're going to get some very unhappy plants that won't germinate and won't grow that well. Ethylene is also what makes bananas mature. If you see plants talking about being gassed, that's often ethylene gas. So that's a really interesting case where, okay, something that you would never think about on Earth is suddenly a factor in space. And when it comes to things like gravity, for instance, water needs to be essentially pushed onto the roots of these tomato plants in a way you'd never have to even consider. So these are just really easy kind of bulk type considerations. You know, everyone in a developmental biology class in college knows about ethylene, except for me, apparently, before 9 a.m. But, you know, basics. So, you know, yeah, I mean, so all of a sudden it just becomes kind of one head scratcher after another. So, you know, the more you can reduce those variables, the better.
Sure.
Yeah.
I mean, I guess there is the advantage. I've heard it read that plants can sometimes grow more densely in zero gravity or in reduced gravity than on Earth. I mean, that kind of makes sense from a simplistic point of view, but is that true? Have you read anything about that?
I have not come across that, but I would not be surprised. I mean, essentially just things are different. That's kind of a short answer, I'd say, is things are different. And definitely there could be things that are better, but any kind of head scratcher in the short term is probably going to manage to confuse a lot of people. So
definitely a lot of research to do.
Yeah, and we'd love if you could send us some link. We'd love to put those in the description so curious readers can dive more deeply. And
I'll definitely hunt down a couple papers for you.
Yeah, absolutely. So one thing we're wondering, and like you mentioned, a lot of unknowns still, but you know, can we expect to be able to grow a complete diet for long duration space habitation using synbio?
Yeah, I mean, that's a great question. I think it all depends on how much space you have and how much money you're willing to put in. The technology, I think, is very close to there for making many things. Biology is so notoriously complex that to say you can make everything is something that I personally would not want to commit to.
Fair, that's fair.
It's just really complicated. But the technology to make many things, I think, is quite there. It's more just how much do you want to pay and how much can you invest?
Yeah.
Right. Well, I mean, and one thing that occurs to me beyond like the idea of growing all your food sounds really appealing, but honestly, if you have to keep some rations, you know, or some micronutrients that are hard to grow in the storage locker and pop one of those every few days, I mean, that doesn't sound like a really bad next best solution, especially if you're sending people to the asteroid belt or Mars, where they're going to be away from Earth for many years at a time. If they can look forward to fresh vegetables and fresh meat, that
and
would
some
be
vitamin C.
Oh yeah.
Right.
And to your point, things like selenium, for instance, metals that are a little bit harder to find for people that turn out to be important for things like mushrooms and some of these other food products, you know, that turn out to be really important for making certain enzymes in people's bodies work. We definitely won't be able to get away without that. But, you know, again, that's something that could be in powder form to add to like a growth medium or a plant medium or something like that, depending on what your setup is.
And I did have a question that we didn't quite touch on earlier. We had talked about using the technology of synthetic biology to make really complicated and important therapeutic materials like anti-cancer drugs. What could you imagine having essentially a pharmaceutical synthesis lab on board that with some genetic instructions that are maybe digitally stored, you can essentially make most of the common medicines that an astronaut would need?
You know,
penicillin and things like that.
I love that question. Yeah, I mean, I think that's possible. It's just, again, like you'd need a crazy set of repositories and gear But I don't think that's impossible. I mean, in terms of complexity, I mean, I'm really curious. I don't know that anyone has tried to synthesize some of these more basic compounds that people make through chemistry in space, because there's definitely a biological
Yeah.
improvement. If you can use biological systems to make these things, you don't need the harsh chemicals, which would be a little bit more dangerous for a closed system, for instance.
Right.
But, you know, I mean, I could imagine a world where you just have a crazy, crazy repository of, for instance, yeast lines or something like that, with all the genetic information and production information, purification information to make all the good stuff.
Well, that's fascinating. Yeah, I was just writing a paper on 3D printing in space for things like spare parts, where if you make an outpost on Mars, you have, you know, a bunch of equipment, let's say, for life support. And if you want to use that equipment for a very long time, you expect some failure, right? Things break, you know, as a mechanical engineer, you know, I'm familiar with MTTF, mean time to failure, which is a kind of statistical measure of how long you expect something to run without breaking. And you could, of course, if it's a short mission, design everything to have a much greater mean time to failure. But if you want to have, say, an outpost that's permanently inhabited, you know, maybe by a rotating crew, but used ad infinitum, you're going to need to fix it. And you can't really ship up a spare part for every single component that could fail, because that is just mass and cost prohibitive. But if you use 3D printing from local resources, especially, but even from an Earth-based feedstock, you could 3D print each part as needed, and all you have is a repository of 3D CAD files that tell you how to manufacture that part. And that's a really fascinating kind of light bulb moment for me. Is you mentioned that a similar sort of repository where you might have all the yeasts that can manufacture the wide gamut of pharmaceuticals that humans might need, but just a little bit of each until you need that pharmaceutical. And then you would grow that yeast into a production colony and make the pharmaceutical and be able to essentially send the knowledge and the information on how to make it without having to actually send the mass of those pharmaceuticals from Earth.
Correct. Yeah. Yeah. And I mean, it just depends how far back you want to go, right? But I mean, yeah, exactly. You know, things like backups, you know, like, okay, it's really good to have that material that you could combine with your 3D printer, right? Which could help you potentially make or purify a lot of those reagents. So yeah, I mean, that's definitely, I think, a really, really interesting thing to think about.
Yeah.
Wow. I feel like we've just taken a big step forward in terms of glimpsing what will be possible with self-sustaining, you know, habitation off the Earth. I mean, we have a big spaceship we all live on. It's just enormously large to imagine building a second one, right? So we have to think smaller going forward. So,
Yeah, and all these technologies can get us less and less reliant on, you know, direct supply from Earth. So
right.
that's fascinating.
Well, Kate, I think yeah, we're kind of running up against the hour, and I just wanted to give you a chance to talk about anything else that we might have missed in this conversation. Anything else you want to bring up as a topic?
I'm trying to think about it. I think you all have done such a great job leading a conversation around this, because I actually didn't realize how far we could take this in terms of depth of what you can do on a spaceship, how you can do it, what the parameters are around that, because that's something that I think is going to heavily govern how we can think about what we can do biologically in space, because you all are pushing the envelope, I think, in a lot of different directions for what's available in space. And once those parameters start changing, you start getting microgravity, you start getting more space, more, I think, light harvest than people have thought about in many other contexts, right? All of a sudden, then, you know, you can think about biology. When you can think about biology, then there's a whole lot there you can start pulling from that, honestly, I don't think has been plumbed that deeply yet. So I think that's the one thing is thank you. I think this has been a really fun conversation that went a lot of places I didn't think we could go.
Oh, I'm glad. Yeah, yeah. I think we felt that way as well. And but yeah, we want to open this. That's part of having this podcast, is to open this this box of thinking further and invite new creative approaches. So,
Absolutely. All right, we got a few minutes left, and we've got one more question. Aiden asks, what are the worries around radiation or other deep space factors that might contaminate on-orbit biological experiments?
Yeah, that's a really good question, because radiation is important for biological systems because DNA can be damaged. So there are a lot of mechanisms that we have in cells that prevent DNA damage. So on the macro level, people have compounds in their skin that prevent, as we know, radiation from getting into skin cells, which are towards the surface of our bodies. And even if you're not talking about things like skin cells, you do have a bunch of molecular machinery within every cell in your body to counter the one-off case of DNA damage from radiation, which will inevitably happen. So there's lots of DNA damage potential, and our body is good at staving off a huge amount of it already. That said, space, of course, ups the ante once again. So there's a whole nother set of considerations that are going to be important there. It's likely that UV filtering would be required for any kind of system that will require light. So I can imagine heavy UV filtering for something like algae to get you exactly the types of light you need and not the other types. Increases in genetic manipulation to increase ability to counter DNA damage, I think, would likely be something that is something people would think about. So how do we increase the number of essentially molecular machines that fix DNA? That could well be a consideration. How do we make proteins that cover the surfaces of our cells that reflect light off, so the cells in and of themselves become more UV tolerant? How do they make their own sunscreen, essentially? Those are a couple of considerations when we're talking about things that can be fully isolated from light. So for instance, muscle cells, or some of these other systems, yeast, E. coli, other good stuff that we can grow in the dark. Those I don't think we'd have affected. It would be more the light sensitive ones. So algae for one, plants for another. So active consideration there.
Oh, but yeah, very interesting.
Yeah, it is. I mean, we're planning to protect humans from excessive space radiation, so we probably should be thinking about protecting our food sources as well, right?
Yeah, absolutely. Well, Kate, thank you so much. We've got to wrap it up here, but we really appreciate you coming. This was a really fascinating conversation, and I know I learned a lot. Hopefully our listeners did too. So thank you for sharing your time and insight with us. And listeners out there, please reach out to us if you have suggestions for future discussion topics, or people you'd like us to interview, or if you want to get on the list to join these conversations live when they're recorded. Just feel free to email us. Our email is ourfutureinspace@orbitalassembly.com. You can also find us on Twitter @ourfuturespace, and we're on Facebook as well @ourfutureinspace. So thank you, Doctor, for coming. We really appreciate it. Have a wonderful week.
Thank you. You as well. Thanks so much for having me.
All right. It's been our pleasure. Thank you, Kate. Bye-bye.